Hey you guys, here is the Photosynthesis Lab. So what I have set up here, let me see if I can turn this for you. I have a beaker with water only and 10 spinach leaf discs. So what I did was I put the leaf discs into this syringe and I sucked up the water like this, and then what I did was I put my finger on the end like this and pulled. And what this did was it used the vacuum to suck out the air in the leaf. So I did that for a beaker with water only. There are 10 leaf discs in there, and I did that in another beaker with sodium bicarbonate, which is our source for carbon. Here we go. So here, I also put in just a tiny bit of soap, which allows for the oxygen to go through the lipid membrane. And so every 2 minutes, I'm going to check in to see if how many leaf discs are floating. And I have one here with no light, just for fun. And so every 2 minutes, what I'm going to do is turn this on and show you the number of leaf discs that are floating for each. Okay, so right now at minute 0, there are no leaf discs floating. I'm going to turn on the light. You can see I have 2 heat sinks, and I will see you guys in about 2 minutes. All right, you guys, so here we are. The pure water with just a touch of soap, there are 0 floating. I don't know if you can see that here. There are 0 floating. Here, at minute 2, we have in the sodium bicarbonate, with just a touch of soap, we have 1, 2, 3, 4 floating. And 1, it's almost going to go, but it's not quite there yet. So we won't call that one yet. So 4. And then in the dark, we have 0 floating. All right. I will see you guys in another 2 minutes. Here we are at minute 4. We have, in the pure H2O, we have 1 floating and 1 almost getting ready to float, but we'll just call that 1 because it's still touching the bottom. In the sodium bicarbonate, we have 1, 2, 3, 4, 5 floating. All right. 1, 2, 3, 4, 5. Yep. Oh, well, one's just starting to float. We'll count that in the next one because it wasn't done yet. It wasn't there when we first started. And in the dark, we have 1 leaf disc that's floating and attached to a big hair. So, maybe something for our potential errors, possible errors. All right, I'll see you guys in a couple minutes. Here we are at 6 minutes. And let me move these. We have in the just water, we have quite a few floating. We have 1, 2, 3, 4, 5 floating. And in the sodium bicarbonate, we have 9 floating. And in the dark, we have still just 1 that's attached to a big hair floating. All right, I will see you guys in 2 minutes. You guys, here we are, 2, 4, 6, 8, minute 8, and we have 1, 2, 3, 4, 5, 6, 7 discs floating, and we have 10 discs floating, so 7 in the pure water, or water with soap, and 10 in the water with sodium bicarbonate and soap. And in the dark, So our dark solution has sodium bicarbonate in it as well. So we have two floating in here. All right, I'll see you guys in 2 more minutes. Hey you guys, here we are, minute 10. We have, let's take a peek here. We have 1, 2, 3, 4, 5, 6, 7, 8 discs floating in the water, pure water and soap. And we have 10, all 10 are still floating in the sodium bicarbonate. And we have 2, there's one almost ready to float, but it's like half on, so we're not going to count it until it actually leaves. I'll see you guys in 2 minutes. All right, here we are, minute 12. We have, oh, let me move these beakers for you. We have 8. There's 1 ready for launch, but it's not quite there yet, so 8. 10 still, and 2. Still 2. See you in a couple. We are at, I think this is minute 14, I think. We are at 9. We're at 10 still. We are back down to 1. There's just 1 floating disc. It is no longer attached to the hair, but it is still floating. Okay. You guys, here we are at, I think, minute 16. We have now reached 10. At minute 16, we've reached 10, and we have been at 10. So 10 for the pure water, 10 for the sodium bicarbonate water. And here we are back to 2. So since we're only going to be graphing, I was just doing the dark for fun. Something we could talk about potentially later, but let's worry about graphing just these 2 in our lab. So our pure water and our sodium bicarbonate. Things to think about: which one is the control? Why? Why is it that the discs are floating? And why is it that the sodium bicarbonate discs were floating more quickly? So when you graph these out, it'll have a steeper curve. And so why is that? Okay. So I'm going to give you guys I'm going to run this again, but I'm not going to video it because it'll be just a lot of work to video that. But I'm going to run it again with a couple of different beakers so that we have some more class data so that we can compare and talk about some class averages. Okay. I hope that you guys are well, and I'll talk to you later.